Hello everyone, my name is Muhammad Saimon Mia. I am a graduate student at Department of Pharmaceutical Science in North Dakota State University. I'm working under Dr. Cjo Matthew. My poster is entitled with Talin-1 promotes fibrosis in clear cell renal cell carcinoma through renin-angiotensin signaling. Fibrosis in the tumor microenvironment is hallmark of almost all solid tumors like clear cell renal cell carcinoma or pancreatic cancer. The tumor microenvironment comprises of various extracellular matrices and growth factors. These components of tumor microenvironment induces multiple cell functions and downstream targets to promote tumor growth through various cell surface receptors. Integrins are one of the family of those receptors. Integrins are heterodimeric cell surface receptors with one alpha and one beta subunit. There is alteration in these integrins expression in various cancer types. These integrin receptors get activated by multiple cytoplasmic proteins like kinlin and talin. The complex between integrin and talin plays a major role in promoting fibrosis. This study focuses on finding the importance of talin-1 in regulating the fibrosis in clear cell renal cell carcinoma. And we hypothesize that talin-1 induces fibrosis in clear cell renal cell carcinoma through renin-angiotensin receptor AGT1 and AGT2. To test our hypothesis, we selected a renal cancer cell line named CACI1. We downregulated the expression of talin-1 in CACI1 cells using shRNA plasmid. The cells were selected using puromycin selection and individual colonies were picked up, which was named as clones. We performed immunoblotting and RT-PCR to confirm the downregulation of talin-1 in the clones. The figure 1A here shows the immunoblotting for talin-1 in CACI1 wild type and the clone cells. 1B shows the normalized expression of talin-1 in these clones using GAPDH as a control. 1C shows the decrease in the expression of talin-1 RNA in clones compared to the wild type, suggesting the decrease in talin-1 expression in clones with the use of talin-1 shRNA plasmid. Once the downregulation of talin-1 has been confirmed, we performed multiple cell functional study to see the effect of this talin-1 downregulation in cell functions. Upon activation through talin, integrin forms basically integrin clusters, which later matures into formation of focal adhesion complexes. Paxillin is a well-accepted focal adhesion marker. So we performed immunostaining to see the changes in the expression of paxillin and phosphopaxillin, which is the active form of the paxillin. We found out that with the downregulation of talin-1, there is a significant decrease in the expression of phosphopaxillin in clones compared to the wild type. Upon treating the cells with phosphopaxillin, the phosphopaxillin were visualized using Alexa Flora 555, and the cell cytoskeleton were stained using phalloidin. The figure 2A here shows the p-paxillin staining, and the figure 2B here shows the merge for p-paxillin and the actin cytoskeleton staining, which clearly shows the downregulation of p-paxillin, which ultimately is the marker for the decrease in focal adhesion complexes. Next, we performed cell proliferation and cell migration assay. To perform the cell proliferation assay, we used MTT assay. And we found out that with the downregulation of talin-1, there is a significant decrease in cell proliferation in 24 hours. Figure 3A here shows the decrease in the cell proliferation in clones compared to the wild type. Figure 3b here shows the decrease in cell migration with the downregulation of TALIN1 in renal cancer cell CACI1. Another important cell function is for spreading of the cells. So we performed cell spreading assay in both collagen 1 and vitronectin to see whether there is any change in cell spreading with the downregulation of TALIN1. The figure 4 here shows the cell spreading in collagen 1, and figure 5 here shows the cell spreading in collagen in vitronectin. Cells were allowed to adhere in for 2 hours in a pre-coated collagen and vitronectin cover slips, and the cells were actin cytoskeleton of the cells were stained using phalloidin. Figure 4a here shows the actin staining in both wild type and the clones, and B basically the merged image of nucleus staining and the cytoskeleton staining. I haven't shown the nucleus staining in the poster. And we found, and we use ImageJ to quantify the spreading area of this wild type and the clones in both collagen 1 and vitronectin. The figure 7a basically shows the spreading area in collagen 1, and figure 7b basically the quantification of the spreading area in vitronectin, which shows there is a significant reduction in cell spreading capability with the downregulation of TALIN1 in both collagen 1 and vitronectin. Another major function for a cell is formation of cell adhesion. So we performed a cell adhesion assay to see whether there is any changes in cell adhesion capability in both extracellular matrix, fibronectin, collagen 1 and vitronectin. And we found out that there is a significant decrease in the cell adhesion capability in both collagen 1 and vitronectin. Figure 8a here shows the decrease in cell adhesion capability in collagen 1, and figure 8b shows the decrease in cell adhesion capability in vitronectin. Once we confirm the changes in the cell functionality with the downregulation of TALIN1, we explored some of the downstream signaling molecules. We performed immunoblotting for AKT, ARC, and P38, as well as their phosphorylated form. We found out that with the downregulation of TALIN1, there is a significant decrease in the expression of phospho-AKT, although there isn't any significant change in the expression of total AKT, which I haven't shown in this poster. Figure 9a here shows the immunoblotting for phospho-AKT, and the quantification of this immunoblotting, which shows the significant reduction in phospho-AKT, is in 8b. The phosphorylation status of phospho-ARC also shows the significant trend, similar trend as the phospho-AKT. We found out with the downregulation of TALIN1, there is a significant decrease in the phosphorylation of ARC 1 and 2. 10A is the immunoblotting of phospho-ARC, and the 10B shows the quantification which confirms the downregulation of phosphorylation of ARC 1 and 2 in TALIN1 knockdown cells. Although we didn't see any change in total ARC 1 and 2 expression, again, I haven't shown it to this poster. We found a contrasting result in case of phosphorylation of phospho-P38. We found out a significant increase in the expression of phospho-P38 in clones compared to the wild type. Figure 11a here shows the immunoblotting for phosphorylation of phospho-P38 and its quantification in figure 11b. Then we perform immunoblotting to see the two major receptors that are involved in renin-angiotensin system, AGT1 and AGT2. We found a contrasting result in their expression. With the downregulation of TALIN1, we found a significant reduction in AGT1 receptor, which is basically the pro-fibrotic receptors in renin-angiotensin system. The figure 11A is showing the immunoblotting for AGT1 receptors, and 11B is the quantification. 11B shows the q-RT-PCR data for AGT1 receptors in clones and the wild type. But in case of AGT2 receptor, we found a significant increase in the expression in clones compared to the wild type. The AGT2 receptor is mainly known as antifibrotic, known for its involvement in antifibrotic signaling. Figure 11C shows the immunoblotting of AGT2 receptor in clones and the wild type, and figure 11D shows the normalized mRNA level of AGT1 receptor in clone compared to the wild type. So to summarize the finding of this poster, we see a significant decrease in both cell adhesion, proliferation, and migration with the downregulation of TALIN1. We also see a significant changes in downstream signaling molecule phosphorylation of AKT and phospho-ARK1 and 2 with the downregulation of TALIN1. But a contrasting result in case of phosphorylation of phospho-P38, where we see a significant increase in the expression of phosphorylation of P38 in clones compared to the wild type. And we also see a significant decrease in pro-fibrotic receptor AGT1 in contrast to significant increase in antifibrotic receptor AGT2 of renin-angiotensin system. At the end, I would like to thank all the funding agency for supporting this research. And with this, I would like to finish my poster. Thank you.